Welcome today to the RNA Collaborative Seminar Series, hosted today by the RNA Research Center in Poznań, which is a consortium of RNA-focused researchers from Faculty of Biology at Mickiewicz University and Institute of Bioorganic Chemistry at the Polish Academy of Science. A few words about RNA Collaborative Seminar Series. This is an initiative working in frames of RNA Society, currently joining 27 institutions all over the world. And you can visit our YouTube channel to find a lot of interesting talks. And today's seminar will be also uploaded there, so no worries. And today we'll have two 25-minute talks. And after each talk, we'll have short time for some questions to the speakers. And please remember to write the questions to the Q&A sections, then I will read them aloud and we will listen some answers from the speakers. My name is Dawid Bielewicz, and it's my pleasure to introduce today's two speakers, Anna Baud and Monika Piwecka. The first speaker is going to be Anna Baud. Anna completed her PhD in structural and functional biology and has extensive experience in mass spectrometry-based proteomics. She is currently a postdoctoral fellow in Professor Sobczak Group at Mickiewicz University. Anna's research combines her expertise in proteomics with RNA biology and neurodegenerative diseases. Her research is supported by Marii Skłodowska-Curie Individual Fellowship and grants from National Science Center in Poland. The second speaker is going to be Monika Piwecka. Monika is passionate about RNA biology for over 15 years. She's a biotechnologist and biochemist by training and molecular and system biologist by passion. Currently, she is a group leader and the head of the Department of Non-coding RNAs at the Institute of Bioorganic Chemistry. So now, Anna, please share your presentation.
Thank you, David, for the kind introduction. I hope what I want to present you today is the project that we started some time ago. And the main goal of this project was to find what drives the RNA translation. And I hope that after today's talk, you will remember that these are the insulin growth factor binding proteins that can directly regulate this non-canonical process of RNA translation from a mutant FMR1 mRNA containing expanded CGT repeats. So the whole story begins with a fragile X messenger ribonucleoprotein 1 gene that is encoded on the X chromosome, as you can see here. In normal situation, in the 5' UTR of this gene, there are CGT repeats up to the number of 45. However, due to expansion of this CGT repeats to the number of 55, or from 55 to 200 CGT repeats, we can call the situation as permutation, which leads to the fragile X-associated syndromes. When the number of CGT repeats reaches over 200, we can talk about full mutation, which leads to fragile X syndrome. The prevalence of this full mutation is about 1 in 4,000 males and 1 in 6,000 females. However, the fragile X permutation is much more common in the society. It is 1 in 800 males and 1 in 200 females. So today I will focus only on the permutation, which leads to the fragile X permutation associated conditions, or FAXPAC for short. So these conditions can be manifested at different stages of life, starting from childhood or adolescence, where they can manifest as neurobehavioral disorders such as learning disabilities, social impairment, anxiety, or seizures. In adulthood, it can manifest as a dysfunction of endocrine and immune systems, leading, for example, to fragile X-associated primary ovarian insufficiency, which leads to premature menopause and issues with conception. It can also cause psychiatric and neurological disorders, such as Fragile X-associated neuropsychiatric disorders, anxiety, depression, or sleep problems. When this permutation manifests during the later stages of life, it can lead to Fragile X-associated tremor ataxia syndromes, or FAXTAS for short. As I told you before, this permutation of CGG repeats is pretty common. However, due to incomplete penetration, FAXTAS can happen only in, is estimated to happen in around 40% of males with permutation over age 50, and 8 to 16% of females who carry this permutation over age 50. So the estimation of FAXTAS in the general population is about 1 in 8,000 for men. So the main features of FAXTAS, so the pathological hallmarks, are is the presence of inclusions in patient cells. In here, I show you the inclusions that were documented in astrocytes, neurons, and Purkinje cells. Also, cortical atrophy is observed in FAXTAS patients, as well as thinned corpus callosum. FAXTAS is progressive neurodegenerative disease that is characterized by action tremor, cerebral ataxia, dementia, Parkinsonism, and brain atrophy. And I hope I could give you an answer about for the question, what drives the FAXTAS, what is the exact pathomechanism, but I'm afraid I will not be able to do so today. Because to date, there are three main pathomechanisms that were described for FAXTAS, and it is believed that they are non-exclusive. So historically, the first mechanism that was described was the RNA gain of function, in a similar way as it was described in other repeat expansion diseases, where this expanded CGG repeats in the 5' UTR of FMR1 gene can form a hairpin. Then this hairpin can attract certain RNA binding proteins and sequester them. This leads to the functional depletion of those proteins from the cell and impairment of their function. So here we are talking about the RNA gain of function. The second mechanism is the excessive R-loop formation, which are RNA-DNA hybrids that can cause DNA breaks and trigger DNA damage response. And the third mechanism that was described over 10 years ago is non-canonical repeat-associated translation, or RUN translation for short. And today I will focus mostly on this mechanism. So as I told you before, the permutation in this 5' UTR of FMR1 gene is caused by expansion of CGG repeats that are shown here in red. And when the ribosome scans during translation through the 5' UTR to encounter the canonical AUG codon, because of the presence of this expanded CGG repeat, it dwells and it stalls and selects non-canonical near cognate codon rather than canonical AUG codon. And when it, for example, for the production here of those proteins, it can select, for example, ACG or GUG codons that will lead to the production of toxic proteins such as polyglycine, polyalanine, or polyarginine. And what is also important to remember is that not all ribosomes will start this RUN translation. This process is fairly not very efficient, because some of the ribosomes can pass through this structure and still start translating from the canonical AUG codon, leading to the production of canonical FMRP protein. However, This process is not very efficient, but these proteins, mainly polyglycine, were detected in FACSTA's mouse and cell models, as well as in FACSTA's patient brains, as you can see on this picture here in red. The accumulation of ephemeral poly-G that is built from the part of the 5' ETR of ephemer 1, as well as the tract of CGG repeats that encodes for glycine. So it is so this protein is built of multiple glycine amino acids, is able to aggregate and form nuclear inclusions in cells. What suggests the role of this process in FACSTA's pathomechanism. And to date, there were several run modifiers of CGG run translation described in the literature, such as the X3X helicase, serine arginine resplicing factor 1, proteasome subunit beta 5, and recently described by us, by our group, ribosomal proteins RPS26 and 25. However, as you can see, the reports about the proteins that can modulate this run translation process are rather scarce. Therefore, that prompted us to investigate this mechanism further, and we aimed to find proteins that can modulate this process. So our methodological approach was based on the RNA protein pull down. And to now briefly, I will describe you this method. So I prepared different RNA molecules which were transcribed in vitro, and that were corresponding to the either RNA encoding 5' ETR of ephemer 1 with expanded CGG repeat, 5' ETR of ephemer 1 but devoid of those CGG repeats. And as a control, I prepared the RNA that encoded a molecule that had similar GC content and length as the first molecule. However, this GC nucleotides did not form hairpin. Those RNA bases were then incubated with a cellular extract, and proteins that were bound to the base were pulled down and identified using high resolution mass spectrometry. Further, some of the best hits were selected for further stratification, and their impact on run translation was measured based on cellular tests. So as you can see here, we using this approach, we managed to identify over 200 proteins that interact with ephemer 1 5' ETR containing expanded CGG repeats mRNA. And those that were specifically upregulated on this expanded CGG repeats comparing to the GC-rich control, there were over 80 proteins, and amongst them we could identify proteins that can be described as RNA binding proteins constituent of ribosomes, proteins playing role in translation or RNA processing. But amongst those hits, we were particularly interested in the protein shown here in red, insulin growth factor binding protein 3. First, we started our investigations by validating the mass spectrometry results. So as you can see here, using orthogonal pull-down technique, we could confirm the binding of ephemer of IGF-2BP3 protein to this RNA containing expanded CGG repeats, but also to the RNA that did not have CGG repeats, comparing to the GC-rich control. Then in the next step, we applied filter binding assay, which confirmed that insulin growth factor 2 binds with a high affinity to the ephemer 1 5' ETR with CGG repeats, but not to the GC-rich control. So having those results in mind, I would like to now tell you a little bit more about this protein. So insulin growth factor binding protein 3, or IGF-2BP3, is a protein that is mostly expressed during embryogenesis, and its expression declines during development. In adult human, it is mostly expressed in reproductive organs, in skin, and inhibitory neurons. This protein is RNA binding protein that has many biological functions depending on its target. So, for example, when it can play a role in transport, stress granulose assembly, translation, or transcript stability. First, having the mass spec data and the biochemical data, we wanted to check whether IGF-2BP3 can regulate the level of untranslated FMR poly-G protein in different cell models. So we started with the cell line with stable expression of polyglycine that was produced by my colleague Izabela Bruniarek. And in this cell line, when we silenced the IGF-2BP3 protein, we could observe also the decrease of FMR poly-G protein. What is very interesting that we also had a cell line stably expressing polyglycine that was produced from a short repeats in the normal length, 16 CGG repeats. And silencing of IGF-2BP3 also impacted the production of this short polyglycine. However, this knockdown of IGF-2BP3 did not influence FMRP level. So then the next step was to do this experiment the other way around. So we overexpressed IGF-2BP3, and we noticed that upon overexpression of this protein, the level of polyglycine increased comparing to control, confirming the possible role of this protein on the production of FMR poly-G. So the Western blot results showed you mostly the data about the polyglycine that is soluble, but as you may remember from the introduction, some polyglycine can also aggregate in cells. So we asked whether this knockdown of IGF-2BP3 will impact the level of FMR poly-G aggregates in cells. And as you can see here, indeed, the knockdown of this protein led to significant reduction of FMR poly-G aggregates in a cell model. And flow cytometry confirmed both the data from the microscopy and Western blot, and showed that upon knockdown of IGF-2BP3, the level of GFP positive signal decreases. Next, we ask whether this effect of IGF-2BP3 is specific to run translation. As you may remember from introduction, this polyglycine is produced from non-canonical ACG codon. So in this experiment, we replaced the ACG codon by canonical AUG codon, meaning that the polyglycine was now produced from the rival Cossack contact, and its basal expression was much higher. However, in those cells that were producing polyglycine from this canonical codon, when we silenced IGF-2BP3, we could no longer see impact of this knockdown of this protein on the production of polyglycine, meaning that this regulation is run translation specific. The specificity was further confirmed by in vitro translation assay performed by my colleague Izabela, who added different concentrations of IGF-2BP3 into the reaction. And as you can see here, that upon addition of IGF-2BP3 to the level of translated FMR poly-G increases, so, confirming its role in translation. So now, then we asked how this IGF2BP3 can modulate this run translation process. And from paper by Schneider that was published a couple of years ago in Nature Communications, we knew that there are some RNA motifs that are recognized by IGF2BP3. And these are CA motifs with a spacer followed by GGC-rich core. And we identify such motif in our plasmids encoding polyglycine. As you can see here, there is CA nucleotide followed by GGC cores formed from, of course, CGG repeats. These plasmids were prepared by my colleague Daria Niewiadomska. And first, I checked whether silencing of polyglycine produced from this plasmid with different reporter leads also to the downregulation of polyglycine level. As you can see, this is the case, which suggests that the observation of polyglycine downregulation upon IGF2BP3 knockdown is not dependent on the attack used for our constructs. So secondly, we mutated this CA nucleotide motif. And as you can see here, when this motif was mutated, the polyglycine could be still produced. However, it was no longer dependent on the IGF2BP3 silencing, suggesting that this may be the site where the IGF2BP3 binds in order to regulate run translation. Finally, we asked whether IGF2BP3 can be targeted by other approaches than small interfering RNAs. And as you may remember, IGF2BP3 is mostly expressed during embryogenesis and its level is rather low in other tissues. However, it's a protein that also increases during cancer. And there are several small molecules that are described as IGF2BP3 inhibitors. These are bromodomain inhibitors shown here. So when we administer those proteins to the cells expressing polyglycine, first of all, we could indeed observe the downregulation of IGF2BP3 protein and mRNA level. But more importantly, these molecules not only inhibited production of IGF2BP3, but also production of polyglycine, as you can see here. And they acted in the same way as silencing of IGF2BP3 because they were not regulating the level of polyglycine produced from canonical AUG codon. So apart from IGF2BP3 protein, there are also two other paralogs, IGF2BP1 and 2, that are expressed in other tissues. And as you can see, their expression is different. IGF2BP1 is expressed also in reproductive tissues, while IGF2BP2 is ubiquitously expressed. These paralogs share high sequence identity, over 60%, and sometimes they even share the RNA targets. So having this in mind, we asked whether other paralogs, namely IGF2BP1 and 2, can also impact the production of ephemeral polyglid from during the run translation process. So as you can see here, silencing of IGF2BP1 and IGF2BP2 resulted in a significant reduction of polyglycine level in the same way as IGF2BP3, as silencing of IGF2BP3 parallel. In parallel, overexpression of those proteins also led to increase of the production of ephemeral poly-G, again in the same way as IGF2BP3 paralog. And more importantly, these paralogs were not able to regulate a level of ephemeral poly-G that was produced from canonical AUG codon, suggesting that these three paralogs act in a similar manner on the biosynthesis of ephemeral poly-G. So with this, I would like to finish my presentation, and I hope that I convinced you today that we identified a novel set of proteins that interact with ephemeral 1 5' UTR RNA, and that IGF-2BP3 protein directly binds to ephemeral 1 5' UTR mRNA, and that all three IGF-2BP paralogs can specifically regulate CGT-run translation, the production of polyglycine, and that BET inhibitors, so bromodomain inhibitors, can diminish the level of both IGF-2BP3 protein and mutant polyglycine. And with that, we can imagine, of course, a hypothetical mechanism that when IGF-2BP3 is bound to the RNA, to the ephemeral 1 5' UTR containing expanded CGG repeat, it adds another level of sterical block for the scanning ribosome, and thus it's increasing the production of ephemeral poly-G. However, in the situation where IGF-2BP3 is repressed, the ribosome can more easily bypass the neurocognate codon, and the ephemeral poly-G production is decreased. And with that, I would like to thank you for your attention, and I would like to thank all my lab, especially my PI Krzysztof Sobczak, and the funding bodies Marie Curie Actions and National Science Center of Poland. And I'm happy to take your questions.
Thank you very much, Anna. So
yes.
Now we will have a short Q&A session. So the first question from Anonymous: did you try to knock down all IGF and BP isoforms, so 1, 2, and 3, to see if the effect on run translation is higher?
Yes, so I did try this once, and because I was also expecting that if I did knock down all of them, I would the effect would be more spectacular. However, it was not the case, but I must admit that I did this experiment only once, and it is my plan to repeat it just to make sure that I didn't have some issues with, for example, siRNA.
Thank you very much. And now we have the questions from Margaret Bomer. Can the CGG repeat form a G quadruplexes?
So we are here mostly talking about hairpins, but I think that there were some reports also saying that G quadruplexes can be formed.
Thank you very much. I cannot see more questions. So now, thank you once again, Anna. And now we'll go to the second speaker, Monika Piwecka. Monika, you can share your screen, and I'm looking forward for your presentation. Monika, you are muted.
Hello, do you hear me now?
Yes, everything fine.
Okay, great. So welcome, good morning or good afternoon, everyone, depending where you are. And it's a pleasure for me to talk about one of the projects that we do at the Institute of Bioorganic Chemistry, Polish Academy of Sciences. And it's going to be also about RNA binding proteins a bit. So give you a bit of the overview what we are doing. So I'm working in the department of non-coding RNA, where we study different non-coding RNAs, such as circular RNAs, long non-coding RNAs, also microRNAs, and we are interested in their cell type specificity, their subcellular localization, and also functions. So what do they do? How do they do things in cells? And as we know, and probably I don't have to tell this to this audience, but RNAs, also non-coding RNAs, are interacting with proteins, and this happens almost through the whole RNA life cycle. So except for looking at non-coding RNAs, we are also interested in RNA-protein interactions. Broadly speaking, in our research, we are looking at the regulatory non-coding RNAs and how do they interact with RNA binding proteins, and we look at this in a tissue and cell type specific context, especially in neural cells, but also in the neuroendocrine cells, such as cells that are existing in the pituitary gland, so the master gland in our body. And of course, okay, I'm trying to move on with the cursor and it doesn't work, so I will have to stop the presentation for a moment. I'm sorry for that. So I don't know what happens, but I couldn't move the slide. Okay. Of course, also our non-coding RNAs, they are important for the regulation of mRNAs. So many times you are looking also at the protein coding genes. And I hope right now you see a slide with the title crosstalk between RNA binding proteins and RNA. And this crosstalk happens all of the time through the whole RNA cycle. And RBPs play critical roles in virtually from RNA birth, from nascent transcripts out until they decay. And they are involved in RNA processing, modification, stability, translation, localization, transport, and so on. And just a short reminder that RNA binding proteins can bind both to double and single-stranded fragments of RNA. And this is quite large class of proteins. There are over 2,000 RBPs that are deposited in databases for mouse and over 4,000 for human. And this binding of proteins to RNA happens via so-called RNA binding domains, and many of them are known. This is the case for RNA recognition motif, K homology motif, and so on. But also many proteins can bind to the RNA via so-called intrinsically disordered regions, IDRs. And what is important for this talk that what has been widely recognized nowadays, that distorted protein RNA interactions are increasingly recognized in different neurological disorders. And also the previous speaker, Anya, was talking about the case like this, but we know also many more examples whereas, for example, mutations in RBPs decrease the expression levels, or we feature aggregation of certain proteins and mislocalization, or aberrant RNAs are sponging or recruiting RBPs so that they cannot do what they normally do, or target the targets that are usually targeted in the healthy cells. All of these ultimately alter RNA metabolism, and this is also widely recognized for cancer nowadays. In my talk, I will be talking about specific neurological disorders, which are associated with demyelination and dysmyelination. And in the beginning of this presentation, I would like to give you a short introduction about this problem. So we can distinguish actually both demyelination, which is a progressive degradation of the myelin sheath, often caused by inflammation that attacks myelin, and there is dysmyelination, so inborn error of metabolism of certain proteins that are building myelin, and quite a number of different neurological disorders, more and less common, are having this kind of myelin problems. And in our studies, we use a mouse model. It's called Shiverer, which is a mouse model of dysmyelination. So there's a single mutation in the MBP gene, which is encoding myelin basic protein. And this is a main constituent of myelin in central nervous system. So this leads to this mutation leads to the failure of oligodendrocytes to form compact myelin in the central nervous system, and this animal have a phenotype that is associated with early tremors that appear in the live seizures, and also this leads to premature death of these animals around 25 weeks of life. So what do we know about RBP-RNA interaction in this dysmyelination or demyelination condition? So actually this knowledge is very limited. There is one lab that is studying this for many years now, and they are made a lot of progress in understanding how RBP called agent RNPA1 dysfunction impacts multiple sclerosis. And they shown very convincingly that a cytoplasmatic mislocalization of mutated version of HNRNPA1 is mainly causing that this mutated protein is actually produced, but it's getting mislocalized to the cytoplasm. So normally this protein is localized to the nucleus, or mainly to the nucleus. However, in the patients, there is this mislocalization that was actually originally found in patients, so in the brains of multiple sclerosis patients, and then these mutations were validated and analyzed more in detail in different cell models. But except for this, we don't know much more. So our goal in this project was to shed more light onto RNA protein interactome in the dysmyelinated mouse brain. For this purpose, we use a method called XRNAX. And here I present a brief scheme how it looks like. It might look complicated and very colorful, but I will try to break it down for you. So this method was originally published by Jeroen Kriksveld lab, and it was used to analyze RNA protein interactome in cell lines and different conditions, stress cells versus healthy cells, healthy and happy cells. And in our hands, we tried to adapt it for analysis of the tissue. So it was a major challenge. We used two different homogenization methods, and brain lysates were homogenates were subjected to UV cross-linking, and this is a very essential step that allows that certain RBPs, RNA binding proteins, can that are interacting at the very moment with the RNA can be covalently linked. Then we use a series of biochemical steps to isolate RNA protein complexes and produce so-called XRNAX extract or XRNAX extract. What do we do with this XRNAX extract? So this extract should be in principle enriched for RNA binding proteins and RNA crosslinks. So first we checked that if and whether we have this enrichment. And indeed, looking at the few different canonical RNA binding proteins such as AGO1, ILAF-L2, which is brain specific, or PTBP2, we observed that the interface samples, so the samples that should capture RNA protein complexes, upon cross-linking, we see that those RBPs are accumulating, and in many cases, these proteins are kind of withdrawal or less abundant in the organic phase sample. We also look at the RNA accumulation in this interface sample, and we used a bunch of different assays for that, mainly qRT-PCR, but also northern blots. And here I show you one result from a northern blot for circular RNA-01 antisense, which is one of the most abundant non-coding RNAs in the mammalian brain. And in the crosslink sample, XL sample in the interface, we see that this RNA tends to also accumulate upon cross-linking. Also, I mean, this analysis helped us to find out which of the homogenization and cross-linking method is most suitable for looking into those interactions, and we choose one of these methods. I will not talk more about it, but we describe it in a preprint so that I will advertise it at the end of my talk. So this is how it goes. So this is how it went for the analysis of XRNA extract that we further subjected to high throughput analysis in a way that first we a bit digested the proteins from these complexes, RNA protein complexes. And then we deposited those complexes on silica columns to eventually digest the whole RNA and analyze only those fragments of the proteins that were directly bound to the RNA, and analyze it with mass spectrometry. And we did it, as I mentioned, for the dysmyelinated brain, shiverer brain, also for the in the brain of the wild-type animal. And in both cases, we discovered that our mass spectrometry retrieved hundreds or over 100 proteins that are mainly RNA binding proteins characterized by molecular functions such as RNA binding, mRNA 3' UTR binding. There are also DNA binders, pre-mRNA binders, and so on. Have also ribosomal RNA binding, because this method also does not distinguish between polyadenylated and non-polyadenylated RNAs. So basically, all RNAs that were cross-linked, or majority of them, should be here. And in both cases, the proteins that we measured are having different non-RNA binding domains, such as RNA recognition motif, zinc finger, homology domain, and so on and so on. But we went a step further, and we look into individual peptides that we discovered or measured, and some canonical RBPs, for example, TBP1 or FAS. And here, we detected peptides that are corresponding to RNA binding domains, such as RNA recognition motif or zinc fingers. And that actually confirms that we really capture these fragments of the proteins that are involved in RNA binding. my God, I cannot move my cursor again. Oh, sorry. I think it's some problem with my Zoom. I'm very sorry, but I will have to start sharing again. I'm sorry for this inconvenience. I'm moving on shortly. Could you see a slide with TDP43 protein structure?
Yes, we can.
Oh, thank you. So just finishing on that, we could identify the peptides that are directly bound to the RNA, and that was a very nice confirmation that these methods really work for us. So what did we discover in the dysmyelinated brain, right? Because that was the main question. So we very quickly realized that in shiverer brain, unlike wild-type brain, we have overrepresentation of certain groups of proteins, for example, cytoplasmatic stress granules, and also a different representation of alternatively spliced RNAs. So here on these heat maps, we put together these two groups of proteins that we focused to kind of look into those two different aspects of that are unstudied kind of in dysmyelination. So first of all, I want to make something clear that the net amount of those specific RBPs that I marked here in yellow that are measured or detected more in XRNAX experiments are not coming from different expression of this protein. So basically, both in wild type and shiverer conditions, these proteins were not deregulated. It's just the interaction with different RNA pools were different. That's why we kind of capture them in a differentially in our analysis. What does it mean? For example, in the case of PCBP1, we see this protein both in shiverer brain as well as in wild-type brain. However, the big different picture that we have. So we discovered that PCBP1 forms cytoplasmatic inclusion in certain cell types in the brain. Here are Mosey cells, and we quantified it to show that there's more PCBP1 inclusions in the shiverer brain, which we are studying more in detail right now. But today, I would like to focus more in detail into alternative splicing and MBNL1, so muscle-blind-like 1 proteins that we show or we validated that indeed known RNA targets of MBNL1 are differentially spliced in shiverer brain as compared to wild-type brain. So we used data to find out about RNA targets and measure this and confirm that indeed MBNL1 works differently or binds differently RNA in shiverer versus normal wild-type brain. However, I mean, the whole thing is much more complicated because also this protein is very complicated. So the gene contains a lot of alternatively spliced exons, and this gene is used as a template for transcription and later on translation of over 10 different MBNL1 isoforms. Also know that MBNL1 locus is really auto-regulating, meaning the protein can bind to the introns of its own gene to influence splicing of its own exons. also from the homolog muscle-blind in Drosophila, we know that this locus can produce circular RNAs that we are also studying in our lab. And this circular RNA can be bound in the cytoplasm by MBNL protein itself. So we look more closer into those non-coding RNA isoforms that, in case of mouse gene, mouse MBNL1, are two located circular RNAs. And what was very interesting that one of these was markedly downregulated in shiverer brain. What is interesting, this circular RNA contains exon 3, 4, 5, and 6. And two of these exons are harboring nuclear localization sequence. Also MBNL2, which gives rise to a few different mRNAs, but also one circular RNA, MBNL2, also perturbed in shiverer brain, mainly upregulated. And what is even more interesting, that looking more into mRNA isoforms, we found that isoform with inclusion of exon 5 and 6, so those ones that are harboring nuclear localization signal, is higher in shiverer compared to wild-type brain. So this led us to hypothesis that perhaps inclusion of those of these exons, exon 5 and 6, may something to do with the exclusion or downregulation of the circular RNA that contains two exons, and this also can lead to the regulation of MBNL1 localization, protein localization. I'm sorry. So downregulation of Circ-MBNL1, we also analyze in the different brain regions separately, showing that or finding out that it's different for different brain regions. So, for example, there's not much different in cortex, but there is quite some difference in striatum, hypothalamus, olfactory bulb. We also analyze it for the skeletal muscle and lung, where we don't see any significant change. And we analyzed subcellular localization of MBNL1 expression in those regions, or actually in many of these regions, but keeping in mind what do we see or what did we observe for Circ-MBNL1 abundance in shiverer brain. And we specifically quantified the signal from the nuclei and the cytoplasm. And in this case, we found out that indeed, in the brain regions such as striatum, olfactory bulb, or hypothalamus, where we observed regulation of the circular RNA, we found also higher or bigger accumulation of the protein in the nuclei compared to the cytoplasm. So there's a difference between a shiverer brain and wild-type brain, the accumulation of the MBNL1 in the nucleus. We also were kind of happy to find that also other labs are working on Circ-MBNL and further looking into how this locus is regulated by circRNA production. And this is the work from Sebastian Gardner lab made in Drosophila, where also different isoforms of muscle blind are producing, are actually auto-regulating production of different circular RNAs in a tissue-specific way, so differently in the brain and the eye. And in case of mammalian system and mice, we can see that this is also dependent on the disease kind of context. So to summarize, XRNAX in our hands looks like nice toolbox for studying molecular changes from the interface of RNA-protein interactions in a high-throughput manner, and is also applicable to tissue samples. Using this method, we provided robust catalogs of total RNA-bound RBPs in a wild-type brain, but also in dysmyelination. And in dysmyelinated shiver brains, we showed that there is an increased association of cytoplasmatic granule RBPs to the RNA, and increased number of PCBP1 bodies in the hippocampus of the myelin deficient brain. We also observed altered alternative splicing of MBNL1 targets, and what we showed also the downregulation of the circular RNA, which is produced from MBNL1 locus, coincides with the enhanced inclusion of MBNL1 exon 5 and 6, carrying nuclear localization signal. And this ultimately also influenced the localization of MBNL1 protein itself. So finishing, I hope I made it on time. I'm sorry for the technical problems, and I want to thank my lab, especially Marta and Veronika, who are the main drivers behind this project, and our collaborators that we have many from many for different projects, but especially Remigiusz Serba and Luisa Stanaszek, who are our main collaborators in this project, and thank you to the audience for listening to me. I'm happy to take questions.
Thank you very much, Monika, for this great talk. Now we have time for short questions. Please write them in Q&A. Currently, I cannot see any question. Still nothing, but if they will appear, you can simply, after this recording, you can still answer to them. So once again, thank you, Monika and Anna, for these great talks, and everybody for logging and listening. Thank you very much.
Have a nice afternoon.